All right, so let's talk about chapter 4. I believe that we opened with a discussion about eukaryotes versus prokaryotes. Yes, does that sound familiar? Okay, awesome. I'm not going to rewrite that long list again this morning. I just wanted to remind you and remind myself and have it on the video that we already did that. So today we're going to start, and did we do flagella? I kind of remember that as well. Yeah, but it was really fast, right? That one, I think I'll do that part over for you real quick. Okay. So chapter 4 is like anatomy of a prokaryotic cell. And I think I reminded you, or I showed you the picture of what I like to call superbacteria, which is in the opening panel of your book. It's figure 4.1. And superbacteria has everything which no bacteria actually has, right? So just keep that in mind, that everything's on that picture in, it's like a good review sheet with a little bit of detail. And just like we did in BIO1, we're going to start with, I'm going to describe the structure of each thing, like span out a protein, carbohydrate, what it looks like, and then we'll talk about the function. Okay. And if I forget the function, just shout out and remind me so that you actually get all the notes. All right. Okay, so let's start with the anatomy, and I think I'm going to start with the outside, work my way in, is usually what I do. So I'm just going to review flagella because I know that we did that super quickly at the end last time. Okay, so the flagella in bacteria is very different than the flagella in eukaryotes. It has 3 parts, so structure first, 3 parts, and those parts are called the basal body, the hook, and the filament. Okay. Most all 3 of these parts are made of a protein that is called flagellin. That's easy to remember. Right? It is not like eukaryotic flagella and cilia. It is not made of microtubules. It is not made of microtubules in a circular arrangement. And because of that, the bacteria are going to move differently through the media than eukaryotic cells. Okay. The basal body is the part that attaches the flagella to the cell, and it's different if it's a gram-positive cell wall or a gram-negative cell wall, so I'm just going to leave it at that. The hook is the part that spins because it's curved, right? It actually does this. And the filament is what you want to think the tail or the extension that actually forces the bacteria through the media, right? So they come in different arrangements. Not all bacteria just have 1. Some have 2, some have 5, some have 50. And where they are positioned on the cell, most of the time, the cell is a bacillus. So what shape is bacillus? Rod, right. So most of the time they're rod-shaped cells, or almost rod-shaped cells. Like you usually don't see a flagella on a coccus, right? And if it's single, we call it monotrichous. If there's 1 on each end, we call it amphitrichous. If there is, I like to say like a tuft or a clump of them on one end, that one we call lophotrichous. And if they are all the way around the cell and they can be of varying lengths, then we call it peritrichous. And if I could, I would show you those pictures, right? But super very briefly, right? Monotrichous like that. Amphi, sometimes it's more than 1 or 2 out to the end. When we look at flagella under the microscope, prepared slides, we're going to look at an amphi and we're going to look at a peritrichous. Lopho kind of looks like this, right? Kind of all at one end, and then peritrichous all the way around. And like I said, they can be of differing lengths, like that. Why is this important? Well, because if we're doing the 5 eyes, there are bacteria that are pathogens that look like this every time. And if you see that under the microscope right away, you're like, oh, this might be blah disease. I'm going to put the bacteria in these kinds of media because I can already be on the way to identifying it and thus treating the patient before we even finish the identification part, okay? So these arrangements are important. You do need to know them. Function of the flagella, we say motility, right? We don't use locomotion anymore. That's a 7th grade bio word, right? We say, and the questions in your lab book are always going to be, is the bacteria motile? M-O-T-I-L-E, question mark. And there are ways to tell that without looking under the microscope and looking for flagella. They do certain things to the food, especially if it's semi-solid media, that you can hold up to light and say, yeah, that bacteria is moving. Okay, super simple. I would remind you that the bacteria flagella spins, and so the bacteria actually kind of moves this way through the media. Eukaryotic flagella move in a wave. Okay, so even if on the surface it looks like the bacteria is moving in a wave-like way, they're actually spinning. We also, we'll also mention run and tumble. Did I tell you this story already? Oh, I told another class this story. This is so cool. So until like the 2000s, we thought that bacterial flagella, it was totally random movement. Because what happens is the bacteria seem to do, just like Leeuwenhoek Saunders microscope, they seem to move in a particular direction for a short amount of time, a couple seconds, and then that's the run. And then it appeared like they kind of, hence the word tumble, they kind of did this. They kind of would spin around seemingly randomly in various directions and then take off again in a sort of coordinated direction. So it would be like run, tumble, run, tumble, run, tumble. And when they tumbled, literally, if you were watching the bacteria with a powerful enough microscope, you would just see them, it looked like they were kind of flopping around and then they would just go off in another direction. So this scientist, this female scientist, like I said, in the early 2000s, was studying flagella and was like, I wonder if it's really just random. So what she did was this really cool experiment in that she put the bacteria in, I'm going to just draw it as a petri dish, although I don't think it was. And she put an increasing concentration of food for the bacteria, which is usually sugar, right, and protein. And then in another petri dish, she put an increasing concentration of an antibiotic. It doesn't matter which one right now. So over here, there was barely any, and then over here was super concentrated. Same with the food, right? She put some bacteria here and said, okay, what do you guys do? Is it random or not random? And what she found out was it wasn't random. When they are tumbling, they are sampling the media. They're sampling around and they can sense increasing concentration or chemical gradients, and they move toward the food and away from the drug, right? So this was like, what? Like for 100 years, we've said it's totally random. We had to change all the textbooks, right? And she got a MacArthur Foundation grant, which is one of those genius grants, like when you discover something that's just like, whoa, right? So I think that's kind of a cool story. So I always tell that story. So like I said, you'll see bacteria with flagella, but you won't actually see them moving. You will just see them on a fixed slide because staining for flagella is very difficult. Usually people break them off. You'll see, you'll be able to tell a little bit of this in that, like I said, sometimes I'll give you a test tube and we'll put some bacteria in there and then we'll look to see if the bacteria moved away from where you put them. And you'll be able to see that visually because you're looking at millions of bacteria at once. But you won't actually, unfortunately, get to see that under the microscope. I'm sure there's cool YouTube videos you can watch. Okay, so flagella, and then I think number 2 is usually glycocalyx, right? Yeah, surface attachment, surface coating. So number 2, and I'm going to put that there. Glycocalyx. So any thoughts? What do you think might be in there? Sugars. Yeah, sugars. Glucose, of course. So it's made of sugars, let's say polysaccharides. Right? And usually some proteins or polypeptides, depends on the organism. I can't give you specific examples. I know you're thinking, whew, one less thing I have to know, right? And that's true. But sugars and proteins, both of which are, I would remind you, are sticky. Think about eggs, think about sugar, right? Just in your regular life. And it's the same for bacteria. Having a coating of sugars and polypeptides makes you able to stick to places where the host may not want you to stick, right? So those are not real erasers. So I'm going to erase these guys. And let's talk a little bit about the glycocalyx. If it is present all the time, and I should say, like flagella, it's a sometimes, meaning all bacteria don't produce glycocalyx, okay? If it is present always, we call it a capsule, because that is a little more organized structurally, like I said, and it's permanent, it's always there. Function specifically of the capsule, if bacteria has a capsule, function is for adhesion. Right, to make them sticky, to stick to things like your teeth, your lungs, right? Your mucous membranes, where the mucus is constantly flowing, but somehow this bacteria manages to stick and cause disease, right? As well as protection. And what they protect against is dehydration. Bacteria need water and food, basically, right, to do their metabolism. And why this is important to have a capsule is because almost all the bacteria that cause respiratory infections, like Haemophilus and Streptococcus, pneumonia and Haemophilus influenza, all those bacteria, they have capsules, which makes them more resistant to dehydration. Achoo! When I sneeze and leave those bacteria on a surface. Right? So they are more likely to be picked up by the next instructor who touches the computer or the keyboard and then puts their hand close to their face, right? Then if they don't have a capsule, they're going to dehydrate and they won't be transmitted. So it's important for the bacteria, right? We're always thinking about what's good for the bacteria, not what's good for the host, you, right? What's good for the bacteria is being able to survive and being able to stick to things, right? Not good for you, right? But good for the bacteria. Now, if the capsule is there sometimes, and we say literally it's loosely organized, which means really it's thicker in some places than other places and might look more like an extension rather than this perfectly round thing around the bacterial cell, then we call it a slime layer. It's one of my favorite microbiology words, although I don't use that one for a password, just in case you're thinking about that. Right, we call it a slime layer, and slime layers help bacteria form biofilms. And this is another like booming area of research in microbiology, because what we found out is just like the rainforest, bacteria lay down, multiple species lay down and interact with each other in a particular environment. And as soon as the first bacteria colonize this and they produce a slime layer, another different species can attach to them and another species can attach to them, and so on and so on. And so sometimes on like medical instruments, think catheter, breathing tubes, right? You can get this whole group of bacteria living there and you can't kill them or get rid of them with maybe one disinfectant or antibiotic, right? Because they're very different organisms, just like there's very different organisms in different layers of the rainforest, right? The insects that live on the forest floor are very different than the ones that are living in the canopy. And so plastic, which we use a lot of, right? If it's not disposable, I always like to use the analogy of Think about those soda dispensers in a fast food restaurant. That's a great place for a biofilm, right? Because there's sticky soda there and any bacteria that colonize and then other bacteria and then other bacteria. Don't freak out, because think about it. You've been drinking soda from soda dispensers your whole life and you haven't died yet. So another place the biofilms are found, teeth, right? A lot of biofilms forming close to your gums. That's why you floss. Right, so to get rid of those, because once the bacteria stick and calcify, they form a biofilm that everybody can join on, right? So just a couple of examples. Again, this is a sometimes. Fewer bacteria make capsules than slime layers, and we're finding like a lot more bacteria make slime layers than we thought. That's probably, I should just leave that there. Okay, 3, one more extension that sticks out of the cell before the cell wall are what we call fimbriae and pili. Fimbriae, or pilus singular, pili plural, right? Same protein, both of them are made of a protein called pilin. Okay, that's pretty easy. Sort of different functions, okay? Fimbriae, we call them fimbriae when there are many, and usually they're a little shorter, not always, shorter than pili. And the fimbriae function to help the bacteria adhere, again, with the sticking. And believe it or not, new data suggests the bacteria can also use them to move. They can also use them for motility. Not like a flagella, like through the media, but more like a slow creep, right? So the fimbriae go down, the bacteria sticks, and then a longer fimbriae extensions and the bacteria scooches. Yeah, isn't that creepy? It's really creepy, especially if they're bacteria that are causing a urinary tract infection, because they're colonizing your urethra and they're climbing ever closer to your bladder, right? And the E. coli that cause the most common urinary tract infections, they're even cooler than this. They have a lot of fimbriae on one end, and they actually, when you pee, the force of the urine, the bacteria does this. Right? And then when the stream stops, the bacteria rights itself and it keeps creeping up your urethra. How cool is that? Right. Again, if you're the bacteria, not if you're the person with the urinary tract infection, obviously. Right? Pili, or pilus, I guess, / pili, those are used for conjugation, which is what we call horizontal gene transfer, but shorthand, if it helps you remember it, you can think of it as bacterial sex, because that you will remember that more than you'll remember conjugation, right? But it's really, it's called horizontal gene transfer. And what that means is, like, it's very descriptive, right? Taking DNA and moving it from one cell to another. Right? So some bacteria have a plasmid, which we'll talk about later when we get to what's in the cytoplasm. And if they have a plasmid, they can make one of these pili and attach themselves to a neighbor, and then they can transmit DNA to their neighbor. And it's usually cool DNA, like, hey, let's be resistant to an antibiotic. I'm resistant, and everybody I can touch is now resistant too. And already you've seen in the lab that the bacteria, well, today you'll see more, the bacteria grow in clumps, right? And they're touching every other cell around them. So once one of them has this capacity and they give it to all their friends, and they give it to all their friends, and they give it to all their friends, pretty soon all the bacteria inside you are resistant to that drug, right? All right, so that's good for those. Number 4. Did anybody print the outline or looking at it? Is number 4 cell wall or cell envelope? Cell envelope. Okay, thank you. So your textbook makes a big deal kind of toward the beginning, you'll see, of this chapter about the cell envelope, or maybe it's those PowerPoints that I can't call up right now because Canvas isn't working. I'm looking, just give me a second. All right, well, they used to make a big deal out of it. Yeah, cell envelope, right? So the outer edge, right, of the bacterial cell. So this may include glycocalyx, cell wall, and then we would also consider cell membrane. Or if you like to call it the plasma membrane, that's fine. I like to call it cell membrane, but you can call it plasma membrane. It might help you remember, right? Different from the cell wall, and what are all membranes in all cells made out of? It's the structure of the plasma membrane or the cell membrane. Very good. Phospholipid bilayer. What did you have for BIO1? Yes. So these are all going to be made of different chemicals, different compounds, different arrangements, right? We've already talked about this one. We're going to spend a long time talking about the cell wall because you might remember from chapter 3, we talked about how the cell wall is what makes the stains different colors, right? And so that's a really important feature in bacteria that we can tell when we're doing the 5 eyes. We can stain and look to see what kind of cell wall you have, because knowing what kind of cell wall you have literally starts which antibiotics you should begin to kill those bacteria, because different drugs affect the different cell walls differently. Okay. And then, of course, we'll talk about the cell membrane and its functions. So like I said, we've already done this one. Check. We're going to spend a long time here talking about cell wall, structure, function. And then we'll talk about cell membrane, see how far we get. Okay, so almost all, never say all forever, right? Almost all bacterial cell walls contain at least some peptidoglycan. Are you in between? The bottom, okay. Like, I just see you squinting at me, it looks painful. Okay, what do you think's in this? What's the first part say? Peptides, which are made out of? Amino acids, good. And sugar again, right? So use your words, right? Use your science words. So peptidoglycan, almost all bacteria have some amount of peptidoglycan in their cell wall. Okay, so we're going to spend some time talking about peptidoglycan. It is composed of just repeating N-acetyl-glucosamine. So it's a modified glucose and N-acetyl-muramic acid. And we are going to abbreviate those as NAG and NAM for short, because we don't want to have to write that all the time, right? And basically, I'm not a great draftsman, but basically, I can draw glucose, I think. So there's the oxygen, right? With some modifications sticking in and out of it. And they're joined together, they alternate. So I should make them a different color. Let's make the NAGs black and pink, green. I know it won't look that different to you, but and the NAM's green. Okay. And they alternate. So that's what I mean by alternate, like this. So that would be G, and then it's attached to an M. Think your book. Okay, that's really bad glucose. So yeah. There. Right. So these molecules alternate to make a chain or chains up. The chains get connected at varying places, but not in between each of them. The chains get connected to form a sheet. So I'm going to redraw this. I know it looks a little more green now, maybe. And I'm going to put a nag in here and attach to another one. Right. So not in between each of them, right? But occasionally there are these bridges. Those are peptides. That's why I made them purple, because in all biology books, proteins are purple, right? So they get connected. So we have chains, and the chains are connected to form a sheet or sheets, and then, or a layer, if you want to think of it. And then the layers are connected to each other with a very specific set of peptides that we call tetrapeptide, because it's the same 4 amino acids that repeat. And I won't ask you to know which 4 amino acids that is. If you can remember tetrapeptide, that's good for you, right? So here, where these peptides are, occasionally there's going to be these 4 amino acids that stick upward or stick downward, and those tetrapeptides attach sheets or layers to actually form what we would officially call peptidoglycan. And the difference between a gram-positive cell wall, a gram-negative cell wall, and an acid-fast cell wall have all to do with how many layers of peptidoglycan are formed and where they are positioned outside in that cell envelope, right? And again, I would show you a picture, but oh, I can't resist. I must show you a picture. Let's see if I can find a picture now. Please, Canvas, please. Kind of thing. There it is. Yay. Yeah, well, maybe that was asking too much. There it is. Okay, so that's a little big, isn't it? Can I make it a little smaller? There we go. So first, I would like to show you this. Can you see that? You can see it, right? There we are. Okay, now the people will be able to see it on the video as well. So these are those different arrangements of flagella that we spoke about. This is with an electron micrograph, the electron microscope. So this is like the most, when we look at it under the bright field, it's not going to look like this. Okay, just be prepared. Those are some fimbriae that are stained with a fluorescent microscope. So fluorescent dyes and UV light, compound bright field microscope. but you can see, I love this picture, right? So the fimbriae are yellow around the edge. The blue, that's like DNA inside. And then obviously the orange is the rest of the cytoplasm. So this looks like some E. coli with some serious fimbriae on them. Here's a picture of a capsule. See the dark stained, so that's the bacteria. The clear area around it is the capsule. And then here is the actual chemical formula of N-acetylglucosamine and N-acetylmuramic acid. That's why I just drew them as basic glucose units on the board, because I wasn't going to draw all the accessories. Okay. Sorry, there you can see that. And then this is what I was trying to show you as far as the chains forming a layer. Notice the yellow connections of peptides. They're not between each of the glucose units, but just sort of randomly between the glucose units. And then depending on how many of those are piled up, that's where we get a gram-positive cell wall versus a gram-negative cell wall. So the picture in your textbook right now in the 6th edition, figure 416, is a lot better than this, but you can get sort of the general idea. So I'm going to redraw this, not as beautiful and without as many colors, right? But I did want you to see that before we talked about it. Oh, I love this picture. So this is an electron micrograph picture of the difference between a gram-positive cell wall. See, it's kind of, this is peptidoglycan. See how it's stained in like brown? And then the yellow is the cell membrane. And then this is gram-negative. See the green and then the brown and then the yellow. Notice how much less brown there is, the peptidoglycan, and where it's positioned. Okay, now I'll put all the words on the board. So erase over here. But I'm going to leave that up now because I went to all the trouble to draw that. So you have to keep looking at it. Okay, so cell wall. Let's go with types. Okay. So first type, we're going to talk about the gram-positive cell wall. I'm just going to abbreviate cell wall now, CW. Okay. So the gram-positive cell wall, that is a super thick, multiple layers with tetrapeptides of peptidoglycan. So I'm going to draw that in black here on the edge of the cell, right? So this is supposed to be the surface of the cell. Thick, multiple layers of peptidoglycan. There are some other things in there. One thing is something called teichoic acid. Most of them have lipids attached to them. So on this little one here, I'm just going to draw and label that teichoic acid. And again, that could be a lipoteichoic acid or just a regular teichoic acid. The gram-negative cell wall looks very different because the gram-negative cell wall, the gram-negative cell wall has an outer membrane. So there is a phospholipid bilayer here. Right, you're okay with me drawing it this way, right? Phosphate heads that are negatively charged, lipid tails that are hydrophobic, want to be near each other, right? So there is a phospholipid bilayer. And then, as we saw in that colored in electron micrograph, then there is a thin few layers of peptidoglycan underneath that. And then there is the normal cell membrane right down here. Oops. Yeah, that's right. Whereas on the gram-positive cell wall, we just have this thick layer of peptidoglycan and then phospholipid bilayer. I should have drawn that here. So this is cell membrane. we call the outer membrane. And in a gram-negative cell, we refer to both that outer membrane and the thin layer of peptidoglycan. That's the cell wall. Okay, so it's kind of a double gram-negative cell wall has two components, the outer membrane and then peptidoglycan. Now, something that's sticking out of the outer membrane in a gram-negative cell wall that's super important is, I'm just going to kind of draw it like this, something called lipopolysaccharide, LPS. Lipopolysaccharide. The lipid portion is called lipid A, and that lipid portion is a pyrogen. So those of you that have already had anatomy and physiology, you know what that means, right? It can go to your brain and reset your hypothalamus, and basically that's why you get a fever when you have a gram-negative bacterial infection. This is why whenever you go to the hospital or the emergency room or the urgent care or your doctor's office and they say, do you have a fever? That's an indicative sign of most likely a gram-negative bacterial infection or a handful of viral infections. Right? So that's why they're asking. If you took Tylenol or ibuprofen or aspirin before you went to the doctor's office, then you're not going to have a fever because that's going to block the effect of LPS. Right? Okay, so then the other major type of cell wall, would be the acid-fast cell wall. Which looks different, still outside of the cell membrane. And most cells that are acid fast, they often have a little bit of peptidoglycan. Often sometimes have a thin, that's a sometimes layer of peptidoglycan. And then what makes them different is in their bring another color, but other than purple, which is not appropriate here. So let's think. Buried in their cell wall, they have this other component. It's called mycolic acid. Mycolic acid is super waxy. I think it's derived from a steroid. And so it resists the staining procedure that we use to stain gram-positive and gram-negative cells. But occasionally an acid-fast cell, depending on how old it is, it might stain weakly gram-positive because of that little bit of peptidoglycan in there. Okay. Some of the plus mycolic acid. And again, why is this important? Because these bacteria that belong to this genus, Mycobacterium, like Mycobacterium tuberculosis, causes tuberculosis, Mycobacterium leprae, which causes leprosy, Mycobacterium, I guess those are the two big ones. They both have obviously an acid-fast cell wall. All right, so function of the cell wall. Before I forgot, and then we're going to talk about the gram stain and how it works because we're going to do that today in lab. But function of the cell wall, before I forget, is to basically protect from osmotic shock. I'll say osmotic pressure. You will remember, of course, the plasma membrane or the cell membrane is a semi-permeable membrane or semi-selective, so things can move in and out. Water can move in and out freely. If I take this bacteria and I just throw it in water and it didn't have this cell wall, it would have a hard time not being destroyed because of the osmotic shock. So this cell wall gives an extra layer of protection, mostly, like I said, to protect from osmotic shock or pressure, barometric pressure, or just gas changes. The bacteria is not and doesn't like to have a full-on surround of oxygen. This helps it survive a little bit longer because the oxygen has to diffuse through the cell wall. Okay, now I want to spend a little time talking about, oh wait, one more. Some bacteria lack a cell wall. They are called, you're not going to like this because it sounds a lot like that. They are called they belong mostly to the genus Mycoplasma. And they have steroids, like a lot of steroids in there, or sterols, we usually call them when they're in this form, an alcohol form, in their cell membrane, both to give them their shape and protect them from osmotic shock. I don't think your book mentions it anymore, but I'm going to just throw it on here. Sometimes bacteria lose their cell wall. They're called L forms. And that's a bacteria usually that is living inside of an organism symbiotically and doesn't bother to make the cell wall anymore. I'm going to put can, but don't make cell wall. Produce, that sounds better. And we know that's true because, like, some of the E. coli, for example, that live in your large intestine, they're L forms, meaning they won't stain gram negative. But if we take them out and we put them in culture, they grow with a cell wall, and now they stain gram negative. So they're bacteria that could make a cell wall, but don't. All right, now I'm going to spend a little time talking about how the gram stain works and how it interacts with these two different types of cell walls. Because, like I said, we're going to do the gram stain in lab today. And then erase this. Just leave the LPS sitting there, but I'm probably going to erase the teichoic acid because I'm going to use the purple and the pink for the stains, because the dyes are purple and pink. Right? Oh, I should have brought my brown. All right, so gram positive, gram negative. I need a little more space there. So the gram stain, as I think we mentioned before, was developed in, I think, the 1880s by a guy named Hans Christian Gram. So that's why we call it the gram stain. And the first dye is crystal violet. Crystal violet is a basic dye. So it has a positive charge. So it's going to be a positive stain, right? You can use it as a simple stain. Stop right here. But most of the time it's used in combination for this differential stain. All right. So crystal violet goes on, and it's a basic, and I'm just going to label these purple circles CV, right? Crystal violet goes on, and like I said, it is basic. It is a basic dye, so it's going to stick to the surface because, of course, this is phospholipid, so that's negatively charged, and peptidoglycan is slightly negatively charged. So crystal violet goes on, but what is different is in the gram negative cell, the crystal violet does not penetrate that outer membrane. In the gram positive cell, the crystal violet can percolate, if you want to think of it like that, down through some of the layers of peptidoglycan. It doesn't just stick to the surface, it actually penetrates through the peptidoglycan. Do you see that how here the crystal violet, if it can't get through the outer membrane, it's obviously not going to get to the peptidoglycan. The second step is to add what we call a mordant. It's called Gram's iodine. mordant, think mortar, right? Like hold the blocks together, hold the bricks together, right? So it helps seal or make that crystal violet permanently in the peptidoglycan. So, and I can use this because it is kind of a iodine. It does kind of turn black, right, when you put it in with sugars. So it attaches to the crystal violet, but do you see how that's still on the outside? It's going to attach to the crystal violet, but here I'm going to get it percolating down in and attaching to these crystal violets down here. And what that does is it makes them too big to get back out the holes that they came in, or the gaps, the spaces that they came in. So they're stuck now. The third step is to wash or rinse with alcohol, because that will remove the dye off the surface. This is the trickiest part of the gram stain for you logistically when you're doing the procedure. And I'll remind you when we go to lab. When you apply the dye is going to come in bigger bottles this time than we use for the methylene blue, big squirt bottles with taps on them. And so when you open it up and you squirt crystal violet all over your smear, great, you want it to cover the edges. Then you're going to rinse that off with water and you're going to squirt the iodine on there. And I'm going to say, just flood the slide with as much iodine as you want, squirt it off with water, rinse it off with water. But when you apply the alcohol, we need to be gentle. Because if we take that bottle of alcohol and squirt it really hard on the top of the bacteria, if we use too much or too much force, we can strip the peptidoglycan clean off. And we don't want that. We just want to take off the dye on the surface, right? We just want to erase this with the alcohol. So I'm going to remind you, this is gentle rinsing. Gentle rinsing. So put a few drops on, rock your slide like a baby, let the alcohol run off, put a few more drops on, rock it like a baby, let the alcohol run off. Okay, that's good. Squirt it with water. Okay, because we don't want to strip the peptidoglycan clean off because we want the gram-positive cells to look purple. We don't want them to stain with the counter stain, which would happen if we stripped the peptidoglycan off. So the last step is a counter stain, and I'm going to use the pink because it shows up better and it's going to look pink, even though, remember, I told you gram-negative cells, we're going to say they're red to help us remember negative and red. The counter stain is safranin, which is a red dye. And we're going to put that on here across the top. And I shouldn't have erased all the crystal violet because the crystal violet has bound so tightly to the peptidoglycan on the surface here that when we put the pink dye on top, the red dye on top, it's literally as a layer on top of the purple. Okay. Then I'm going to rinse with water again. This one will come off with water. DH2O for distilled water, right? I'm not just using tap water. And then what's going to happen is this is going to stick and this one's going to come off. And I'm going to have one set of cells that are purple because they retained the crystal violet, and one set of cells that are pink or red because the crystal violet was removed and we added that counter stain that's red. Okay. When we go to lab today, you're going to have 2 gram-positive bacteria, the 2 that should stain gram-positive, and 1 that should stain gram-negative. Okay, and I'll let you sort out which one's which when we actually go to lab. Okay, so today, instead of making 2 smears on your slide, I would encourage you to make them small and make 3 smears on your slide. Right, and I would put, well, I'll tell you what order to put them in that'll help you view them at the end. So I'm going to add something to the function. I'm going to put shape here or morphology on there as well. Which brings us to a good time to talk about morphology versus arrangement before we try to talk about the inside of the cell, which your book did a little bit earlier. So questions, comments, worries, concerns about this so far? Yes, no, no, yes. Okay. And again, I know we talked about this in lab, but we should talk about it again because it's sometimes a difficult thing for people to kind of wrap their heads around. So the morphology does simply mean the shape. And for bacteria, they come in 3 shapes. And I don't want to see anybody who writes sphere or round on morphology, I'm going to mark it wrong because remember, poof, you're all microbiologists. I want you to use your science words. Okay. So bacteria come in basically 3 shapes. They come in coccus, right, or cocci, and that is a sphere shape or round, right? They come in bacillus. I can't spell that right. Bacillus or bacilli, and that would be rod shaped. And those rods can be various. They can be, sometimes we call them pleomorphic because some of them are going to be perfectly shaped and some of them are going to have like a constriction in them. And some of them are going to be more constricted like this, right? Spheres, that's pretty obvious, they're round. Third possible shape, really we call it curved or spiral shaped. And there are 3 subcategories of spiral or curved. A, vibrio, and that literally means comma shaped. So, you know, technically it's a rod, but it has a little curve to it. And all of them would have that shape, right, like that. Spirillum, that's the one you saw the first time we looked under the microscope, right? 1 or 2 curves, gentle curves. Right? And the third type of curve is something called a spirochete, C-H-E-T-E. And that is literally a corkscrew shape, which when you look at it 2-dimensionally, looks like a zigzag. Okay. We're not going to see any of those. Technically, there are also bacteria that we call square. We're not going to see any of those. They don't cause disease in humans. But those are the 3 shapes. So when it says morphology, you're going to write coccus, bacillus, or one of these words, vibrio, spirillum, or spirochete. When we look at the prepared slides of bacteria that have capsules and the bacteria that have flagella, you'll be asked for morphology. And then on the flagella one, it'll say flagellar arrangement, meaning those other words we talked about today, monotrichus, peritrichus, lophotrichus. And then other bacteria, it'll just say arrangement. And I would remind you that the arrangement that's how the bacteria are grouped, meaning in relationship to each other. And the choices for arrangement are going to be single, which is pretty obvious, right? Just one. Diplo whatever, Diplococcus, Diplobacillus, right? Because diplo by itself is just a prefix to mean 2, right? You can't just use that one. You only use that if they're all in pairs. All right, that's a very specific arrangement that you only see in a very few genus of bacteria or genera of bacteria. So if you see some of them that are in pairs, but most of them are in chains, you say they're in chains. Okay, so other choices are Staphylo, and that's always going to be Coccus because rods can't really arrange in clusters because they're not round. Okay. And again, Staphylo by itself isn't a word you use to describe arrangement. You have, that's just a prefix. So we say Staphylococcus. If they're in chains, notice lowercase, Streptococcus or Streptobacillus or bacilli or cocci. That means in a chain, like this. And a streptobacillus would be, sometimes the chain is bent because they're living in a group, right? But that still is streptobacillus. If the bacillus are like this, that doesn't make them a spirochete or a spiral, that still makes them streptobacillus. Okay, so in a chain. I think Bacillus megaterium, which we looked at in our simple stain, is usually streptobacillus, but not always. It's okay if you just put bacillus and you put single, that's fine. Oftentimes, the name of the genus of the bacteria tells you that, obviously, right? When we look at Staphylococcus epidermidis, right, the shape, the morphology is coccus, and the arrangement is Staphylococcus with a lower s, lowercase s. So again, I just thought it was a good time to bring that up. These guys, the spirals, they're always single, right? Because they're curved, they don't tend to form chains or clumps. They just stay by themselves. All right, what have I got for time? Okay, let's talk a little bit about the plasma membrane or the cell membrane. Finish off that cell envelope before we talk about what's in the cytoplasm. Okay, so I'm going to erase over here. And I think you told me cell envelope was number 4, right? I would say A of cell envelope is the cell wall, right? Or if you want to be super technical, you could say the glycocalyx. B is the cell wall. And then the last part of the cell envelope is the cell or the plasma membrane. You already marvelously told me that the structure of the plasma membrane, right, is a phospholipid bilayer. I would remind there are proteins in that bilayer, right? And I would just, keep reminding you things from BIO1, right? Some of the proteins are transmembrane or integral membrane proteins. Some are peripheral in that they're only on the inside or the outside. They don't go all the way through. Some of these proteins are involved in transporting molecules across the membrane, facilitated diffusion, right? Some vaguely familiar. Some of them are receptors to grab stuff going by. So let's just remind phospholipid bilayer, hydrophilic, hydrophobic, right? Hydrophilic heads, hydrophobic tails, plus proteins, and also usually some kind of sterol, right? So in human or animal cell membranes, we talk about cholesterol because that's what's in our cell membranes. In plants and in bacteria and fungus, they're different sterols, but they have them too. And that helps with the structure or the rigidity of the cell membrane, so it isn't just lipids and super squishy. Functions of the cell membrane, still to be a selectively permeable barrier. You gotta have a cell membrane to be a cell. You gotta be a cell to be a living thing, right? I would remind, water flows through freely through there. Gases, oxygen, carbon dioxide, nitrogen also flow freely through the membrane. And then obviously other molecules like organic molecules need to be transported. REQ for require transport across the membrane, right? Second function of the plasma membrane in bacterial cells, in prokaryotic cells, and this is huge, which is the whole reason I'm making a big deal out of the plasma membrane. It is the location, or you'll hear me use the site, the place, for, let's say, metabolism. So if the organism is going to do cellular respiration, which bacteria do, if the organism is photosynthetic and it needs a place to put its photosystems because it does not have a chloroplast, so it needs a place for chlorophyll, right? And needs a place for the electron transport chains that are going to move those electrons and help turn that light energy into ATP and NADPH. It needs a place for the Krebs cycle, a place for the conversion of pyruvic acid to, all right, acetyl coenzyme A. It needs a place for the electron transport chain. This is where it is because it's the only membrane they have. And if you can think about it for a minute, well, now it's really handy if they have a cell wall, right? Because they can move things across that membrane to make those hydrogen gradients, those proton gradients to make ATP, right? Because there's a place where those hydrogen atoms can go and not just float out into the food or out into the water, but they're bound inside. Well, I erased my picture, but they're bound inside. There's a space between the cell wall and the cell membrane, and that's where they go. And then they get pumped back in through ATP synthase to make ATP, right? So those things, what some of your favorite chapters from BIO1, chapter 8 and 9 particularly, are coming back to haunt you soon in a classroom near you, right? Those are our chapter 8. It's all compressed into one chapter. All right, so I'm going to stop here.